welcome to another lecture of the course Aquaculture. Today we are going to talk about farming of pearl oyster Pinctada fucata, the culture methods. Culture methods of Pinctada fucata, the pearl oyster, we are going to discuss in two lectures. So, first, the part A of culture method we will take up now. Pearl, we all know, is very precious and is used widely as an ornament. Highly shining globular concretion found within the shell of an oyster, that is collected from the oyster. That is why these oysters are also referred to as pearl oysters. The idea of the pearl industry was, evoked in Japan uh, for the first time, and it is supposed that Tokishi Nishikawa um, was the person, the researcher, the scientist who first, um, you know, got the spherical artificial pearl. And still in Japan, the pearl farming or pearl culture is very popular. The high quality pearls are obtained from pearl oysters of genus Pinctada. This genus Pinctada fucata is known for high quality, extremely high quality pearls produced worldwide. Pearl oyster aquaculture is a rapidly increasing multi-million dollar sector that many of us know in several countries, tropical marine environments, including, you can see here that how far it is, popular. Australia, French Polynesia, pearl oyster aquaculture, I am talking. Pearl oyster aquaculture is popular in these countries and many other regions that are not listed here. Australia, French Polynesia, the Middle East, China, South Asia, and Japan. There are many regions where these Pinctada fucata are found, and if there are culture technologies that can be applied in those regions, the pearl oyster aquaculture is being undertaken. Japan and China are two primary producers of cultured pearls. I'm talking about Pinctada fucata, and these two countries contribute quite significantly. Now, how to do oyster farming? You know, uh, first we need to have an idea that which species we can culture and how we can culture. What are different procedures? What are protocols that we can follow for culturing these pearl oysters? Now, site selection. The ideal site is sheltered bays. We can have sheltered bays, but there are some other methods also that people, if you are not having this kind of sheltered bays, you can go for other methods also. Or within the sheltered bays, you can adopt different kind of techniques. Culture structures, we can use rafts and cages, and these rafts and cages in different forms, different varieties of different techniques, modifications of these you will find in different regions, and they are very successful in culturing these pearl oysters. Shallow coastal water with a calm environment, rough seas must be avoided. We should select uh, calm seas or calm environment where these Pinctada fucata is supposed to be cultured. Places with cold water currents, there are some more conditions which we need to avoid, like places with cold water current, it has to be avoided. Red tides, again, to be avoided. Pollution by industries, polluted areas need to be avoided in order to save these pearl oysters. And domestic waste should also be avoided. So, in general, they need clean and calm environment that you will have to provide, and especially the shallow coastal region with clean and calm environment. If you have these conditions, then it's wonderful. What are different environmental conditions that we should take care of? Like in Japan, 20 to 25 degrees centigrade is the temperature range where they culture these oysters. and then 23 to 27 degrees centigrade in our country in the Gulf of Kutch during the winter, of course, when we have this kind of temperature. The problem is that if the temperature goes down from the optimal range, say for example 13 degrees centigrade, the oyster may hibernate, or 6 degrees centigrade, if you lower it down to 6 degrees centigrade, the oyster may die. And if it goes beyond 28 degree, then again problems come, and oyster gets exhausted. Ultimately, all these conditions need to be avoided. Try to keep all the physicochemical parameters within the prescribed optimal range. Salinity, not clearly investigated, but prefers high salinity, uh, what is found in the published text. 24 to 50 ppt salinity for a short duration of 2 to 3 days, and 14 ppt and 55 ppt, you know, sometimes 100% mortality may occur at these salinities. So this also need to be taken care of. In fact, all the water quality parameters you will have to take care of by going through the biology of this, and what are different parameters, what is the optimal range of different parameters. All these things you will have to take into consideration. Bottom should be gravelly, depth around 15 meter, silt load. Uh, you have to prefer clear water. That's it. Then oyster supply. Collection, natural beds or reared from naturally collected or cultured spat. So you can have either from wild or from a place where it is being reared. In the Gulf of Mannar, several pearl banks are distributed off Tuticorin at a distance of 12 to 15 kilometer and depths of 12 to 25 meter. Pearl banks, because in these places you get to see these pearl oysters in large number, large quantity, and that Gulf of Mannar is quite rich in this, and you will find people collecting by hand. Scuba divers are also there who go and collect, and they have their bags attached, or baskets attached. With the help of knife or chisels, they take it out and put it into their basket or bag. So collected by scuba diving by hand from the intertidal regions can be collected. In Japan, the collection is done by the divers, particularly women divers are there, called as ama, which means girls of the sea. So they are engaged in collecting the pearl oyster from the wild. They collect oysters with the help of a small hand net and stock them in a wooden bucket, which is attached to their left wrist by a cord. So people have different kind of techniques, so that when they dive, then in minimum of time they can harvest maximum of these pearl oysters. In Japan, during the peak larval settlement season, oyster spat are also collected by submerging bundles of cedar twigs near the water surface. The best time to dive is early morning to midday in 2 months of the summer season, when the water is calm. Hatchery cultured seeds of oysters are helpful and provide the industry with a more dependable source of oysters. It is always recommended that whenever we have to use any candidate species for aquaculture, here in this case Pinctada fucata, we need to have standardized hatchery protocols, or we need to have controlled breeding programs where we can ensure the supply of the young ones for the culture purpose. So that is dependable, that is written here. That is dependable source. So or if you are depending on from the on the wild source, then it's not sure that what you are getting and how much you are getting. For a proper supply of oyster seeds, the artificial incubation of eggs solves the problem of obtaining oysters for pearl culture. So there are different techniques that people have developed over the years, and if that is used, we can get seed for the pearl culture. Now, coming to how to solve this problem and how to get supply of seeds from the hatcheries, we have to maintain, the first and foremost thing is broodstock maintenance. We have to maintain the broodstock of Pinctada fucata or pearl oyster. The broodstock development, breeding, and larva rearing of Pinctada fucata has been done by CMFRI, Central Marine Fisheries Research Institute, and you will find you know the kind of technologies they have developed, very easy and successful technologies you will find with the people from CMFRI. Brooders collection, wild stocking can be done in 1 ton capacity tank, 750-liter water at less than 25 degrees centigrade. They should be fed with a mixed culture of algae, mostly Chaetoceros, at the rate of 4 liter per oyster per day. That is the way we calculate that how much has to be fed. For maturation, supplementary feeding by raw corn flour at the rate of 30 mg per oyster per day is effective. That is how we calculate the ration in this case, and you will have to follow the protocols to maintain the water quality. If you are feeding from outside, supplementary feeding is provided, then you will have to be very careful regarding the organic load or obnoxious gases and other things. Feeding continues for 45 days and mature oysters have a chance to spawn with 30% response. So, if you are doing it properly, if you are feeding them with the well balanced diet prescribed to brooders, then you can get spawning easily. Then these broodstocks can be induced to spawn in the controlled environment. Spawning naturally are induced by chemical or thermal induction. Chemical induction can be done or thermal induction can be done, or they can spawn naturally as well. How we do chemical induction? We can use hydrogen peroxide, tris buffer, or sodium hydroxide and ammonium hydroxide. These are the agents which are used for chemical induction in order to get Pinctada fucata breed. Then thermal induction is also one of the methods, most suitable method. This is considered better method than the chemical induction. If oysters kept at room temperature of less than 25 degrees centigrade in air conditioning room, just listen carefully. If oysters are kept at room temperature of less than 25 degrees centigrade in air conditioning room, if you have kept in the room in the whichever controlled environment where you are maintaining the temperature 25 degrees centigrade or less, when they are quickly transferred to water having 5 to 6 degree higher temperature, from 25 degree or little lower, you transfer them to maybe 30 degree or 31 degree or just less than 30 degree. In fact, we should avoid going beyond 28 degree. So, maybe if you are at 22, 23 degree maintaining them, or 20 degree you are maintaining them, and 5 to 6 degree higher temperature water you shift them there, then that can stimulate spawning. So, this thermal induction seems easy and there is no involvement of any chemical. First, males respond and spawn. Here the things are little different and peculiar. So, it is not like both the sexes are releasing their gametes simultaneously and fertilization occurs. Males respond first and spawn. Due to the presence of sperms in the water, the female gets stimulated and spawns within 30 minutes. So, male spawns first, releases its sperms, and that gives a kind of inducement to the female, which can spawn within 30 minutes of that. Eggs are pear-shaped, pyriform in shape, and about 73.9 micrometer along the long axis and 45.2 micrometer in width. That gives them that peculiar shape. Oysters were removed after spawning. They have to be removed when the gametes are there, when the eggs are there. When you have the fertilized eggs, then the brooders are removed. For 2 hours, water should be undisturbed so that fertilization occurs, uniformly, and good success rate of fertilization percentage is achieved. Fertilized eggs settle at the bottom, and these fertilized eggs now become spherical in shape. Earlier they were pyriform in shape, but now they have become spherical in shape, fertilized eggs. So that way you can differentiate also the fertilized eggs from the unfertilized one on the basis of just shape. Eggs can be collected by filtering the water through a 25 micrometer nylon bolt sieve and washed in filtered seawater. After 45 minutes of fertilization, eggs, that means fertilized eggs we are talking. After 45 minutes of fertilization, early embryonic stage development starts. Within 20 hours of spawning, eggs metamorphose into veliger stage, veliger larvae. Veliger larvae average 62.5 micrometers. Now, larval rearing is the critical part, which is the critical part in many of the aquatic species that we use in aquaculture. Now, larvae should be transferred to larval rearing tanks, and stocking density should be 1 million larvae per ton of seawater. So, we can stock about 1 million larvae, 10 lakh of larvae in 1 ton of seawater. On alternate days, water should be exchanged in order to keep the water quality in check. The microalgal cell Isochrysis galbana that we discussed earlier also is provided to the larvae from the veliger stage onwards. The survival of larvae is very critical in most of the aquatic species, and that has to be taken care of in terms of whether water quality or feeding or food type or choice of the food. So, these things should be done very strategically and with utmost care. Aeration is required after this setting of the pearl oyster larvae. Aeration, proper aeration, thorough aeration must be given so that there is proper growth and no mortality. Here you can see the feeding rate at each larval development. It has been summarized here beautifully. First day, veliger, we feed them 5,000 cells per larvae per day. On 10th to 12th day, umbo stage, we have to feed them 10,000 cells per larvae per day. So, from 5,000 cells per larvae per day, we have now reached to 10,000 cells per larvae per day. On 15th day, when eye spot stage is there, 15,000 cells per larvae per day. Up to 60th day, spat requires 50,000, I should have come this way. From eye spot stage, when pediveliger stage is on 18th day, we have to feed them 20,000 cells per larvae per day. On 24th day, spat, 25,000 cells per larvae per day. 60th day, spat, we have to feed them 50,000 cells per larvae per day. Then up to 90th day, 2 to 3 mm spat, mixed culture of Chaetoceros can be fed and then transferred to farm. So, it's not that strictly only this much of this is an average of feeding rate or feeding schedule that has been given to you based on what is available at your place and how the larvae are responding, and that way you can manipulate the feeding strategy also. Nursery rearing, spats obtained from wild or hatchery should be stocked in outdoor nursery tanks. Stocking density is 5,000 per square meter. Water exchange at the rate of 10% daily. 10% of the total water, if you are replacing daily, then that's enough at this stage, at nursery stage. Feeding a mixture of Chaetoceros and Isochrysis and Nanochloropsis is considered better during the nursing stage or nursery phase. Feeding of algal cells till the oysters reach adulthood, more than 45 meter, you have to use 75,000 cells per ml, right? So, now when adulthood is attained, reduction in stocking rate should be done to 50 to 60 oysters per square meter. Now, when you have grow outs, then how to rear the grow outs? The aim is to grow the nucleated oysters for pearl production. In India, 3 types of farming practices are carried out: rack culture system, raft culture system. Rack culture system in most areas of shallow depths ranging from 2 to 5 meters, one rack of 30 square meter contains 100 cages. So, you will find different type of strategies. It's not like only one type of rack culture system is there that has to be followed by everyone. If you will visit different regions, different places on the same coast, you will find different variations, and that depends on the existing conditions, prevailing conditions in that region. So, raft culture method is also there. The rafts are almost square in shape, and farming is carried out in sheltered bays with considerable depth. In the beginning, I told you that sheltered bays are considered ideal or very good for culture of these. Raft size is 6 by 5 meters, floats with 4 biosphere floaters. Then the third method is long line culture. This is carried out in the open sea with more depth, but usually we should avoid the rough seas. It can withstand high wind and wave action and is hence most suitable because of being in the long line. It can withstand agitation in the water, in the open ocean. But usually rough seas are avoided. You don't have to do it in the rough seas. So, little wind and wave action, that is good, but not rough seas. Cage rearing of mother oysters. Size of the box cage 40 by 40 by 15 centimeter. Mesh size depends on the size of oysters, of course. Frames of cages 6 millimeter mild steel rods coated with anti-corrosive paints or coral tars. Cages with frame of 60 to 40 centimeter and many compartments, 5 compartments, and hinged at one end as open book. They are also used. I told you there are different variations, different modifications of these systems which can be used for culturing the Pinctada. So, this is just one of them. You can have many other type of such systems which you can follow, modifications of such systems which you can follow. Onshore rearing technique, this was developed in India at Visakhapatnam. It's cemented in cemented tanks of size 250 to 500 square meters. Seeds of 5 mm size were reared. Water level was 1 meter. A grid system was constructed using PVC pipes to hold oysters above the bottom. They are not placed at the bottom. In fact, you have to hang them above the bottom. So, for that you will have to make provisions. Here PVC pipes were used for that purpose. Stocking density 125 per square meter. Feeding, as we have discussed earlier also, Chaetoceros is one of the favorite food items of Pinctada. 75,000 cells per ml is the concentration that you have to offer to the Pinctada. Water exchange, here you have to exchange about 25% daily. The water exchange, that how much you have to do, that again depends on the water quality in your region and how quickly the water is getting spoiled, or many other factors that you can work upon for your region, for your site. Average growth 50 mm in 6 to 7 months you can get. Reported growth rate that has been reported that in southeast coast of India, we have seen that there are reports that first year, in 1 year, the growth is dorsoventral length has been taken in millimeters, 45 millimeters. In second year, 2 years of Pinctada, 50 mm, 3 years, 60 mm. Vishakhapatnam, after 1 year, 56.17 mm, and after 2 years, 72.11 mm. Again, Vishakhapatnam onshore system, 3 years, Oyster is 105 mm. Gulf of Mannar, after 6 years, only 75 mm. Gulf of Kutch, after 7 years, 86 mm. Arabian Sea, after first year, 62.5 mm. So these are the reports which are available, but you can see here that quite large variation is there in different places and different regions, and that is understandable because the conditions, the food quality, the food quantity, and the other conditions which are influencing the physiology of the Pinctada, that may be responsible for this varied growth. So that's it in first part of the culture of Pinctada fucata. Thank you so much.